that has to do with cells. And this unit is no different. We are going to talk about how cells grow and the life cycle that they move through. Just like we have a life cycle, we start out as babies, toddlers, kids, preteens, teens, adults, even older adults, right? Like we've got that kind of life cycle going. Cells also have a life cycle. My goal for you for today is to be able to explain to me what limits cell size, so what keeps cells from growing bigger and bigger and bigger, and to name the phases of the cell cycle. That's our goal. So we're going to start off with those limits. Cells can't be the size of a tennis ball, right? Yes. Oh, did I forget your notes? Sorry. Cells will keep growing and keep growing until they reach a certain size. And once they reach that certain size, the cell gets a signal that says, okay, it's time to start dividing. Right? It's time for me to split in half and split into two. Cells undergo asexual reproduction, if you remember from the very, very beginning of the year. And as cells get bigger and bigger and bigger and bigger, they face two big problems. These two things right here, DNA overload and material exchange backups, so like backups to the system, getting things in and out. Those two things are the reason our cells have to divide. So if the DNA starts getting overloaded, or if it's hard to get things in and out of the cell will be like, okay, it's time to split. I've gotten too big. We need to chill. Right? Those are our two limits to cell size. Okay? And we're going to talk about each one independently. So DNA overload, material exchange backup. Clogs, stuff like that. Right. So the first one is DNA overload. As the cell grows, the stuff inside the cell needs to be told what to do, right? The cell needs to have instructions. But if the cell gets too big, the DNA doesn't have enough information to tell that big of a space what to do, right? DNA doesn't grow, right? As a cell gets bigger, the DNA stays the same size. So it's kind of like we've got a good, 20 people in this room right now, right? It's not too bad. I've got enough information. My voice is loud enough to carry throughout the room, right? If we were trying to do this in the cafeteria, right? And the cafeteria was full of people, odds are you wouldn't be able to hear me, see me, get the information that you need to carry out through your day or to reach our objectives, right? So as the cell gets bigger and bigger and bigger and bigger, the DNA, that information, isn't enough to keep the whole cell going. So that limits how big a cell can be. The second is backing up or kind of a clogging of the cell membrane. Our cells need food, need water, and need to get rid of wastes, right? Just like we need to breathe and eat and get rid of wastes, our cells need to breathe and eat and get rid of wastes. The only way to do that is through the cell membrane, right? We talked about that. Endocytosis, exocytosis, molecular transport, bulk transport, right? Ringing some bells from last week. Okay. So the membrane's the only way stuff can get in and out of the cell. The membrane is like a highway in and out of a city, right? And if the cell's pretty small, there's no worries about there not being enough room on the membrane to get out, right? It's like comparing a city like Crawfordsville with Indianapolis, right? Unless there's a train or a piece of farm equipment, it doesn't take too long to get out of town, right? You're going north, you're going south, you're going east, you're going west. There are enough side roads. There's enough space on the highways. You can get out of the city if you need to, right? Relatively quickly. Anybody ever tried to get into or out of Indianapolis during rush hour? That is not a fun time. This right here is what the city of Los Angeles looks like darn near all the time. Right? Big cities have problems getting people in and out of the city, right? Big cells have a hard time getting stuff in and out of the cell because there's not enough space on that highway to get out. So if stuff starts getting backed up, if stuff starts getting clogged up, our cell has to divide in order to make itself smaller. The fancy scientific way to explain this highway metaphor is surface area to volume ratio. You didn't expect me to surprise you with math this morning, did you? Okay. Math and science are best friends. We'll do a little thing. Have you talked about ratios in math? Yes, we know what a ratio is. Sometimes we see ratios written out like this, right? That's a 2-to-1 ratio. So a lot of people, when they like make recipes for like cakes and stuff, it's like there's a 2-to-1 ratio of flour to sugar. So for every 2 cups of flour, you get 1 cup of sugar, right? That's a ratio. This symbol right here is a very fancy way for us to have a division. So sometimes when they ask you for a ratio, right, it's just one number over another. Right? So if we want to find the surface area to volume ratio of a cell, we would take the surface area and divide it by the volume. Making sense? You okay? This is what this chart is saying right here. Our cells want a high surface area to volume ratio because they want a high amount of roads out of the cell based on stuff inside of the cell. That's why me and you, big organisms, are made up of a bunch of little cells instead of one big cell. Because right here, this little cube, we'll say it's like a 1 inch by 1 inch by 1 inch cube. If we found the surface area of it, right, that's 6 because there's 6 1 by 1 surfaces. If we found the volume, that's 1 by 1 by 1, length times width times height, right? And our surface area to volume ratio, or our surface area divided by our volume, is 6. Right? Let's say we wanted a bigger cell, that's 5 by 5. Right? We could add up all of the faces of our cube, get a surface area of 150. 5 times 5 times 5 to find the volume is 125. Our surface area to volume ratio is 1.2. That is way lower than the 6 that we had with this small cell. Cells want to keep their surface area to volume ratio high. So what they do is instead of being one giant 5 by 5 cells, it's a bunch of 1 by 1 cells that take up the same amount of space. So we've got 5 of these 1 by 1 cells by 5 of these 1 by 1 cells. If we added up all of the surface area of all of those cubes, we get 750. But our volume is going to be the same as this big cube right here. When we do our little division, we get back up to 6. This means our cells have a way to get things in and out of them. Doing okay? That's all the math I've got. All right. Couple other little background things that we're going to talk about before we get into the depths of the cell cycle. We're going to talk about chromosomes a lot. I don't think we've said the word chromosome in this classroom yet. That's exciting. When we talked about cell organelles, we talked about the nucleus, right? And that's where all of the DNA is held. Sometimes, not all the time, but sometimes our nucleus needs to wrap up our DNA to make it not get tangled up or not get broken. Let's see if I still have these. Aha. So, let's say I've got some old-timey, maybe to you, wired headphones, right? If you've had headphones like this before, I assume you have. Let's say that you are in a rush. Your teacher tells you, hey, put your headphones away. You just grab them out of your ear and shove them in your pocket, right? Odds are, when you take them out of your pocket, they will be a tangled mess, right? They're in knots. They're broken. It's not easy for you to get them back to where you need them to be. But if you wrap them up in a nice little package, I learned how to do this when I was in like 8th grade. It saved my life. If you wrap them up in a nice, neat little package and you put them in your pocket, they're going to be that way when you take them out. That's how our nucleus keeps track of our DNA, right? It doesn't want it all just tangled up in a big blob of spaghetti. When it needs something, it knows exactly where and how to find it, right? It wraps DNA up around these special proteins called histones. And those things together make a chromosome. And this is kind of what a chromosome looks like. We'll talk about those in a lot more detail when we talk about mitosis later on. Doing okay? All right. So, we're going to talk about the cell cycle. Just like we have a life cycle of baby to toddler to kid to teenager and on, our cells have a cell cycle, and it's got 4 phases. The goal at the end of the cell cycle is for a cell to split into 2 cells. We call those daughter cells because they are 2 independent, like, offspring of that initial cell. And there are 4 phases: G1, S, G2, and M. Those are the 4 phases of mitosis. Not mitosis, of cell division. Sorry, getting ahead of myself. So, during our cell cycle, our cell will grow, ready itself for division, and then divide. And at the end, we'll have 2 little daughter cells, and those daughter cells are going to go right back to the beginning of the cell cycle. Okay? So we're going to work step by step by step, and we're actually going to alternate between the notes portion and that big diagram you have on the very back of your notes. So we might be flip-flopping a little bit. Just prepare for that. Okay. Our first big phase is called interphase. Interphase is just what we call a cell when it's not dividing. If a cell is not dividing, we say it's in interphase, right? G1, S, and G2 all together make up interphase. Okay. Later on next week, my goal is for you, good morning. Later on this week, next week, my goal is for you to be able to identify what phase of the cell cycle or of mitosis a cell is in just by looking at it. But that's for a little bit later. So our first big phase of our cell cycle is interphase. So looking at our big cell diagram right here, I want you to flip back to it. Find it? Should be this exact thing. What I want you to do is draw a line here and draw a line here and connect them. Just like that. That we are going to label interphase. Remember, you can use your notes on any test we take. I highly recommend that this is what you get down. All right, that's interphase. So interphase is made up of G1, S, and G2. We'll start with G1. G1, the G stands for growth. Originally, it was called gap, and I'll tell you why. Back when we were discovering the cell cycle, our microscopes weren't very good, right? When we looked at cells in G1, we thought that they weren't doing anything. So we said, okay, this is a gap phase where nothing happens. Over time, what has happened is we have seen more and more detail in these cells. We have gotten more and more information, and we now know that in that gap phase, that phase where we initially thought nothing was happening, the cell is actually doing most of its growing. So we call that first phase gap 1. This is where it does most of its growing. So what you're going to do is right here, you're going to write G1. This is where the cell does most of its growing. That's gap 1. What comes next? S. Not quite. It is what comes next. But there's a little side street that we got to go down first. Y'all talked about it a little bit in your cell cycle introduction. That was G0. Sometimes cells don't need to divide, right? Just like sometimes people don't want to have kids. That's all right. That's perfectly fine. Sometimes cells don't want to divide. When that happens, they go into a special G0 phase. There are certain very special cells in your body that are always in G0. Okay? Your brain cells and your heart cells. Those cells don't divide because they have very important jobs to do, and they don't have the extra resources to do all of that division stuff. So sometimes cells enter this G0 phase. And the way we're going to draw that is off of G1, we're going to write G0. Because sometimes a cell grows up and doesn't need to divide. That's why brain injuries and heart injuries are so hard to heal. because those parts of our bodies don't like duplicate their cells. And a lot of what healing is cells duplicating in order to fill in gaps. But if those cells aren't able to divide, it's really hard for that tissue to heal. S stands for synthesis. DNA synthesis. Let's say you've got the instructions on how to. Apparently, I'm in a cakes and cookies mood because the only thing coming to mind is bake a cake. Let's say you have a very, very good recipe for a cake and you bake it and you bring it to school and your friends are like, hey, that was phenomenal. I need to make this. Can I get the recipe? Right? You can't just hand them the cake and expect them to know how to make it, right? You have to give them the instructions. You have to give them the recipe. So when a cell duplicates, right, when a cell divides, each one of those daughter cells needs a copy of the instructions, needs a copy of that DNA. So we have to duplicate it. We have to make a second copy of DNA in order for both of those cells to have a copy. So that's our S phase. S stands for DNA synthesis. Let's try not to write in cursive. DNA synthesis or DNA replication. Early on in our next unit, we'll get to learn the cellular mechanisms that allow DNA to replicate. It is a phenomenally interesting process. I can't wait to talk about it. Yes. About? Those cells are allowed to replicate. They grow up and then they stop. Yep. Only some cells don't make any more. Got something to add? Brain cells, yep. Your brain is very flexible and it tries very, very hard to regain those connections, but it'll never be the same. Mm-hmm. That's why heart replacements are a big deal, is because when people get heart injuries, oftentimes the only way to fix it is with a heart transplant. Yeah. Yeah. Yep. Your skin cells are different. Your skin cells are very, very good at duplicating. Your skin cells are duplicating all the time. Half the dust in the room is skin cells that have sloughed off because your skin cells are just duplicating and duplicating and duplicating. Some cells are really good at it. Some cells aren't. After we have our DNA made, we go into a G2 phase, and originally it was called GAP2, but since then we have figured out that the cells are doing a lot more than just sitting around in G2. Right? In G2, that's when we're duplicating our organelles, right? If we're making two cells, we're going to need two different rough endoplasmic reticulums. We're going to need enough smooth endoplasmic reticulum. We're going to need enough Golgi. We're going to need enough mitochondria in order to keep both of those cells moving. So G2 is kind of our last step. Okay, let's make sure I have everything that I need before I divide. So in G2, we are duplicating organelles. get everything ready to go for the big show. That's G2. We got one last phase. What is it? M phase. Our M phase is cell division. Cell division is two processes: mitosis and cytokinesis. I always think cytokinesis is a fun word to say. M phase is mitosis and cytokinesis, and there's a very important difference between these two processes. Mitosis is the division of the nucleus, and cytokinesis is the division of the cytoplasm. A lot of people will use the phrase mitosis instead of M phase. So they will say mitosis is the whole process. But mitosis is just the division of the nucleus, and cytokinesis is the division of the cytoplasm. Together, they make M phase, or cell division. They can overlap as mitosis is finishing up, cytokinesis kind of gets started. But mitosis has to be complete in order for cytokinesis to be complete. We need two separate nucleuses before we can get that cytoplasm to cover up. So, right here we've got our M phase. Whoop, I'm still in highlighter mode apparently. M phase. Our M phase has two parts: mitosis and cytokinesis. We write mitosis like this because mitosis itself has 4 other phases that we're going to talk about next week. For now, we're just going to finish out this diagram by writing PMAT in these little boxes. We'll talk about what PMAT is next week when we talk about mitosis. And once we divide, as you can see here, we've got a brand new cell. What's it immediately go into? G1! And over, and over, and over again. That's a cell's life cycle. Questions? All right. There is an exit ticket on Canvas. Nothing crazy, like 4 questions. You have plenty of time to do it. Once you finish up those exit ticket, make sure that you've finished up the cell cycle activity. You now know all of those steps. So if you don't know an answer, you can look it up in your notes. And then if you're